Hello, good afternoon. It's 4:10 today, so I think it's time to start our webinar for today. Thank you very much for joining us. My name is Natalia Loaiza, and I work at Surgentis as Sales and Account Manager. And I'm very, very happy to be here today doing this webinar for you. I'll be presenting an overview of our TLA technology and how it can be of use in cell line development for biopharmaceutical applications. And be aware that we will have some time for questions at the end, so please send us your questions using the chat tool, and we will try to answer as many as possible by the end of the presentation. But if we don't have time for your question, we will contact you through email to provide an answer. And I like to start with this slide to tell you a bit about our why at Surgentis. Surgentis, it's an international company where all of us come from a biological background. And as biologists, we cannot avoid but keeping the central dogma of biology very present in our minds. And this means that we are very aware that for any cell system to work, and for this cell system to deliver high quality products in the form of a protein, of a peptide, for example, we need to make sure that we are starting with the right DNA sequences for the right genes, ideally also at the right places of the genome. And we understand how crucial this is to make possible new advances in the biopharmaceutical industry. And that's why at Surgentis we are committed to improve the quality of genetic research by enabling cost-effective, complete sequencing of relevant genes using TLA, of course, the technology that Surgentis has been developing and refining since 2012. And we believe our services directly help to improve the efficacy and safety of pharmaceutical cell lines. And we like to think this is the way we can contribute to make the world a better place. And here you can see a typical workflow of the cell line development process. And you know this very well because you are the experts in the field. And you know this is very long and labor-intensive process where proper quality controls are crucial to ensure long-term success. And this happens because, well, genetic engineering is a highly stochastic process in which the scientists in charge can control some of the variables, but not all. So a lot of things can go in unpredictable ways. And that's why the process relies on very robust screening and QC steps in every single phase. And this cannot be underestimated in our competitive world because it can really make the difference for the long-term success of a project. And the quality controls not cover cover cell phenotypes like viability and production titers, but looking at the genetic level is actually a requirement. And then this genetic characterization is usually performed at different stages, so in the master cell bank or later in the working cell bank or the end of production to demonstrate genetic stability. And yeah, this is not only because of scientific interest, but it is absolutely required by regulatory bodies. And these are a few characteristics that scientists are usually very interested to look at. So they want to look at the transgene or vector, and they want to know the specific integration sites in the genome, the integrity of the transgene, the copy number, and all of that information is used to demonstrate clonality and genetic stability, as I mentioned before. And the techniques to look at this are varied. And the idea is, of course, what I mentioned before, to ensure quality and consistency if they produce biological. That's the main goal. And going to the techniques that people usually use to assess all these different genetic parameters of interest. You are familiar, likely, with all these methods. And typically, none of them can produce enough data to comprehensively assess each of these genetic variables. So for example, fish can be used to study integration sites, but it doesn't tell you much about structural variation, co-integrations, or single nucleotide variants. And Southern blot, well, it can tell you about rearrangements, but it doesn't tell you about the other parameters. And so you will need a combination of approaches to get all this overview, and that ends up being, well, require a lot of time and also a lot of resources. So fortunately, there are new alternatives like TLA, where in one single assay, you can gather information, very comprehensive information to assess the complete genetic makeup of your cell line. And well, it ends up being more cost effective when you can just use one method instead of a combination of methods. And because TLA has shown to be accurate and versatile, the technology has received a wide endorsement in recent literature, and not only in academic research, but mainly by pharma and biotech companies. And for the specific application of cell line development, we already have 10 publications, peer-reviewed publications. But if you consider all the other different applications like cell therapy, genome editing, or transgenic animal models, then we have way more publications. And for those of you who are not familiar with TLA, well, now I would like to give an overview of the technology and the types of data it generates. And starting from the basics, the short form TLA comes from targeted locus amplification. And as the name indicates, this is not a method to look at the whole genome. This is not a genome-wide approach, but TLA allows to study specific loci of interest with very, very high resolution and including the surrounding genomic context. So TLA is based on the principle that neighboring nucleotide sequences are by default also close to each other in the 3D complex configuration of the chromatin. And that's why TLA starts with a first DNA cross-linking step, where all the nucleotides that are in close proximity in the chromatin, they become enriched. And in this way, we also enrich for nucleotides from the same locus. And after cross-linking, we do DNA fragmentation, gene restriction enzymes, and we religate all these fragments to obtain circular structures of DNA. And this reshuffling of the DNA occurs in a stochastic manner in millions of cells in parallel, which gives a broad coverage of the gene of interest as well as the surrounding. And then we use a special TLA primers that are located outwardly, and in that way we are able to reconstruct the integration side of the transgene in a hypothesis-free manner, meaning that we don't need any a priori information of the genomic location to be able to determine the integration sites of the transgene. And this way, well, as I tell you, we can give you integration sites, we can give you data about structural changes around the integration site, vector integrity of the host genome. And something very interesting is that this is a typical TLA that you would obtain from a TLA experiment. And here it has this typical bell-shaped curve, covering from 50 to 100 kb, and then you have increased signal from the point of anchoring for the primers, and then the signal decreases with distance. But still, we have very high coverage all over this peak, which allows to look at the sequences and the structural variability with very high very high accuracy. And to tell you a little bit more how we collect our data and how the TLA experiment is designed, is very important to mention that for our standard TLA analysis, we use at least 2 pairs of unique TLA primers, and we locate them at the a little bit in the periphery of the vector, let's say, the extremities of the integrated sequence, one primer here and one primer pair here. And this yields 2 complete independent data sets that allows to perform cross-validations between data sets. This means that each integration site and sequence variant will be internally validated by the assay itself. And so that generates very strong data for your experiment. And if you had vectors that are larger than 50 kb, so 2 pairs of primers are usually enough for 50 kb. But if you had a larger vector, that's not a problem. We just include additional primers. That is very common, for example, for back inserts in mouse models. A little bit less frequent in cell line development projects, but just for you to know, in the case of TLA, well, the size doesn't matter. And TLA data is then mapped to the vector reference sequence. And this type of data is represented in this type of plots. You will find this in all TLA reports. And what I want to show you here is, well, here is the location in the vector sequence in the x-axis, and in the y-axis we have coverage, which here the axis goes from 0 to 1,000, but actually here the axis is cut. So actually we have way more reads. We are just showing you at least 1,000 reads. And TLA will yield very high coverage across the integrated vectors, as well as the adjacent genomic sequences. And what it shows blue is that you have full coverage in all these points of the genome. And this is an example for a 10,000 base pairs vector. And you see around the middle that there is a little bit of white. And these white areas suggest that there is a little bit less coverage than compared to these other areas of the sequence. But still, we have at least 500 reads in this area, which is still very good. And these white areas indicating reduced coverage, like it's a drop in coverage. This can happen because it corresponds to the primer binding sites, or it can also be due to intrinsic limitations of the next generation sequencing, for example, difficulties with CG-rich areas. But as I said, well, it's not really a problem because we still have enough coverage. And with this very high coverage and multiple data sets, we can ensure that we can reliably call each sequence variant that you find in a TLA report. And now moving on to when we map TLA data into the genome home, the genome of the host. Here you will find always in a TLA report these genome-wide coverage plots, where in the x-axis you have the chromosomal position, and in the y-axis you can find chromosomes for mice, but for CHO cell line, well, the genome annotation release still doesn't reach the chromosomal resolution. We usually work with the Cree PICR CHO genome, and well, still doesn't reach chromosomes, so we work with scaffolds. And when we map PLA data in these plots, you will see an enrichment where the transgene is integrated. For the rest in the genome, you just see like the background. And the peak of TLA here, it shows a very nice single integration site. That happens with targeted integrations, but if you will have a random integration approach, you will find the peak in different areas, and it doesn't matter where, PLA will be able to find the location. And well, this is very important to find the existing integration sites. And because it's a hypothesis-free approach, you will be always able to define the location in the genome. Let's continue. So in addition to these genome-wide coverage plots, when we zoom in one specific integration site, TLA will generate high sequence coverage across these breakpoints resulting from the integration. And we will give you these sequences with single nucleotide resolution. So you can exactly pinpoint integration. And similar when you have multiple copies of the transgene or virus, we will be able to tell you the transgene fusions, which allows to detect partial deletions or integrations or rearrangements, or the transgene copies. And this information is essential to pick up potentially truncated copies of your transgene, which might lead to downstream expression of aberrant proteins and might generate complications for your final product. So this is very important data. And just to give you an interesting example of the level of genetic detail you can find with PLA, using this type of analysis. This study, together with the JAX laboratory from a few years ago, we were able to spot that several transgenic mouse lines contained contaminating bacterial sequences inserted in the genome, which had co-integrated with the transgene. And this is and this has been reported for mice, also for cows, but also in cell line development projects. And this is just a very interesting study case that kind of shows that you can always discover this type of surprising events only by looking at the genome with a very powerful unbiased method like TLA. And besides genome transgene breakpoint sequences, in the case of a structural variants within the vector or multiple vector copies integrated at a single site, TLA will similarly pick up vector-vector breakpoint reads indicating this. And you might be aware that the process of DNA integration in the genome is very stochastic and variable, and it's very common that the vectors do not integrate in single copies, but they form concatenaires. And these, they fuse to each other, producing vector-vector fusions. And not all of them are always in the same configuration, so you will have this vector, let's say, in the right configuration while the next one is flipped. So you will find this tail head, like let's say here is head-head fusion, here we have a tail-tail fusion, and you can imagine all the possible configurations. And this is important because it can result in aberrant proteins and might interfere with the quality of the final product. So genome vector fusions are also very important. And this also has been recently reported in the literature that this configuration can have influence in whether the transgene will be more effectively silenced in the long term. So if you have too many tail head fusions, you will have steric restrictions for the access of the transcription machinery. So that's something interesting to keep in mind, and that's why researchers are usually so interested in looking at detail at these confirmation and configurations of the concatenaires, and they are interested on resolving the concatenaire. And well, with TLA we can provide quite some data to help in that regard. And so TLA is also very useful to study large structural variation that can happen in the transgene or also in the host genome during the genetic engineering. And as I mentioned before, we can look at the TLA peak, and when nothing wrong happened, when everything went as expected, you will find this bell-shaped curve. But when we have a structural variation, we see deviations from this pattern, and we can verify this visually. For example, when we have an inversion, instead of having a complete bell-shaped curve, you have two halves of the curve, looking like this. When you have a deletion, you will have a very clear gap where the sequence is missing. And the same with translocations. You will see one side of the peak in one chromosome and the other side of the peak in another one, and so on and so on. And the presence of this structural variance can also be confirmed based on vector-vector breakpoint reads. So overall, we will give you very detailed information of whether any structural variants are present in your cell line. And because TLA is coupled with next generation sequencing, of course, it will provide data with single nucleotide resolution. And for example, here, compared to the reference genome, we have the presence of two common frequent variants. So, for example, here, all the reads show this C, and all the reads here show the G. And here, well, we have a little bit less coverage, but overall the data is very congruent. And this is for the case of frequent variants, but if there were any infrequent variants, TLA would also be able to detect them. And here we will be able to tell you which percentage of the total reads is supporting this. So here we have approximately 20% of the reads showing the A substituting the G, and this will give you, it's a reflection of the bulk sample, and unfortunately it doesn't tell you anything about a single cell. And all this single nucleotide data is reported in convenient tables where you know which primer set is supporting the mutant with how much coverage, percentage of the reads, etc., and you can even compare between samples. So this is a summary of all the TLA outputs that you can get in one single experiment. And all of these outputs are very important genetic features, very relevant for cell line development projects. And this is a lot of information in one image, and you might not be able to read in detail what is there in the text, but I just want to summarize that TLA allows detailed characterization of transgenes and provides information on integration sites, concatenaires, breakpoint sequences between vectors and the host genome, and also structural rearrangements, co-integrations, sequence variants, frequent and infrequent. And all of this in one single experiment, which makes TLA a very ideal methodology that would be an ideal choice for the QC of multiple genetic engineering applications. Well, that was the overview of TLA, and of course, all of this shows that it's a cool technology, but I bet you are already asking how can we specifically apply it to support cell line development projects. And this is what I would like to talk about in the next part of the presentation. And here I aim to cover the TLA applications for cell bank characterization, and as well as for the assurance of monoclonality and genetic stability. So as this is again the initial image I showed summarizing the cell line development workflow. And throughout this process, we already discussed that QCs are very important. So that's why we are able to support our customers at multiple stages. So initially, in early stages, TLA, it can be used upstream for process optimization, for example, to evaluate and QC new technologies. So for example, new transfection methods, or if you are testing an artificial promoter to enhance expression of your transgene, well, we can check that everything is in the right place. We can identify integration hotspots, the integration sites and whether they are clean or not. We can tell you if the integration happened in target or not. And we can also provide genetic data to study and compare high producing clones versus low producing clones. And we can analyze both monoclonal samples and also heterogeneous pools, so that's not a problem for TLA. And that's a point where we can help you, but we more typically, we talk to customers who already perform their genetic engineering, and now they are screening and selecting their top candidate clones, which have the desired phenotypic characteristics. So at this stage, well, the same data that TLA can produce can be used for select or deselect siblings. Of course, you want to make sure the clones you are selecting don't present any unwanted variation in the gene of interest, especially in the coding region. You can use TLA data to select the cell lines that present the optimal copy numbers of the transgene, also in the ideal places of the genome. And of course, for research purposes, TLA data allows to understand the genetic basis for phenotypical differences between low and high producing cells. And I will provide a few examples from the literature for these specific applications for cell bank characterization. And this comes from a publication from Novartis in the Biotechnology Journal, and they provide examples on how they use TLA specifically for deselecting siblings. So TLA helps determine which candidate clone shares the same integration site with others, and therefore they probably share a genetic origin. And what you want to do is you don't want to select too much for clones coming from the same origin. You want to ensure diversity in the different candidates. So in this case, the genome integration sites for clone A and B are identical, both in the chromosome 2, suggesting they are siblings. So it means one of these clones should not be selected, while the others that show genetic the diverse integration sites, they are ideal to be included in the screens. And this is one example from the literature. Another interesting example from the literature showing how TLA data allows to understand the genetic basis of phenotypical variation between clones. Here, this is a paper from Novo Nordisk and the Technical University of Denmark and the University of Copenhagen. And here they use a combination of approaches to study their clones. So they use RNA-seq and TLA to study the phenotype. So one clone is high producing while the second clone is non-producing. And the RNA-seq shows a very complete transcription pattern in clone 2, but not for clone 4. And TLA was able to explain these phenotypes. Basically, clone 1, as expected, shows very clean, intact sequence, while clone 14 has a large truncation in the promoter. So this is very interesting for research purposes, or to answer diverse research questions in this stage of the development. And finally, well, after the cell line has been selected and banked and perhaps cultured, often regulatory authorities require the complete assessment of clonal derivation and genetic stability over time. So we also help in that phase. But before I go to give you more examples of that, we wanted to share with you that, well, TLA, as I mentioned, it can be applied to study random integration events, but also single events or multiple events, multiple vectors. And here we see that there is a shift from the random approach towards more focusing on targeted integration using landing pad methods. And this is an interesting shift because it seems that companies have the need to have more control over the genomic location at the transgene of the genome. And this happens because, well, actually, the integration site seems to have a very strong effect in the genetic stability of the line. And that's one of the conclusions of this recent paper published last year in November. It's a paper from Janssen and the University of Natural Resources and Life Sciences in Vienna. And they study retrospectively the factors that influence genetic stability in CHO cell lines. And actually, well, the first one identified is the location. So good choice of integration site is critical. And they, for example, they mentioned that intronic sites seem to be more favorable than intergenic sites. And they add a number of other parameters that are a little bit better understood. So for example, integrity of the transgene and concatenarization pattern is very important, copy number, and also the presence of genome rearrangements. And with TLA, we can help looking at several of these genetic parameters. The third factor that was found to be very relevant was related to culturing conditions and stress signals. And of course, that's not a genetic factor. So in that sense, TLA is not relevant at all. But for the other two, for sure, our services will be able to cover you in your projects to ensure all these parameters are in the right place according to what the literature has already found. And for cell line development projects, we work both directly with the companies developing the cell lines themselves, but also with CDMOs. And I wanted to highlight that last year we had this publication with Fujifilm from UK. And here we show that the Fujifilm has integrated TLA in the Apollo X platform. And this has helped increase the speed of the projects and also to improve the quality of the end product. And these CDMOs tell us that their clients very frequently ask to include TLA as part of their service. So it goes both ways. And just for you to know, TLA is also available when you work with a CDMO. And of course, I just want to highlight that for our cell line development applications, we can work with CHO, but we can also work with any other cell system, even yeast. If that were something that you are working on, please feel free and we can share our experience with the different models. And well, finally, as I mentioned, some people reach this final state when they need to go for registration into the authorities. So in that stage, it's mandatory to provide data to prove monoclonality and to prove genetic stability. And in that area, we have developed new packages to support the projects. And that's what I would like to cover now. And well, let's start discussing why is monoclonality so important. Well, a very well characterized cell line and cell line bank is very, very fundamental to ensure the quality of the products, of the therapeutic products of the industry. So assurance of clonality, it's key to ensure consistency and quality and the performance of the product during the life cycle. So that's why it's mandatory. And the FDA has recommended to use 2 rounds of limiting dilution at sufficiently low seeding densities to provide acceptable proof of monoclonality. However, today the industry has moved to use a combination of approaches, for example, equipment based on flow cytometry, single cell sorting, which is automated and also coupled with cell imaging systems. So now there are multiple ways to prove clonality. But, well, many programs still work with legacy cell lines that were created way before this advanced industry equipment was available. So in that sense, having a method that shows clonality using genetic approaches is also very interesting. And that's why we have worked in developing tools to help the industry in this regard. So we are now offering a service package that takes care of providing the data to prove clonality based on the work published with Novartis. And last year, we released the white paper describing this approach, which consists of 2 parts. So in the first part, we analyze the master cell bank together with divergent clone, unrelated clone, and we performed a complete genetic characterization using 4 sets of primers and extended sequencing. And based on the breakpoint sequences obtained with TLA, we then designed primers to perform a specific qPCR analysis in 93 clones. And with this, we can provide data that confirms monoclonality with 95% confidence. This is published, so if you would like to take a look at these documents, we can share them with you. Please feel free to request them via email. And this is the summary of the data that we first generate for the master cell bank. And based on this, we then design the PCRs and the qPCR primers. And then we do the subsequent analysis of the clones. So that's the second part of the report. And very frequently, some of our customers also like to perform TLA in some subclones, but this is optional. And now moving to the genetic stability package, the FDA requires providing data from at least 2 of these 3 cell banks. But most of our customers typically analyze these 3, the master cell bank, the working cell bank, and the end of production. And similarly to what we do with the clonality study, we first perform TLA on the master cell bank to determine the genetic makeup of the line, and subsequently we analyze the downstream clones. And if they yield exactly the same integration size, the same concatenamere patterns, and the same mutation profiles, we can deduce that the cell line is genetically stable. And well, with this genetic assessment based on TLA, we can provide comprehensive and reproducible data that summarizes the complete genetic makeup of the cell line, and we provide detailed documentation to support this. And of course, all this data builds and provides very strong insight into the genetic stability, which gives a lot of confidence that actually the biologicals are of high quality. And TLA data has been used as primary data in regulatory applications, and this is a frequent question we get. And of course, I hope you understand that we cannot disclose too much of these details because it goes into the confidentiality agreement with our customers. But of course, TLA data is favorably reviewed and included as primary source in the applications. And just to summarize, so TLA can be used in multiple stages of cell line development, including clone selection, cell bank characterization, providing genetic data about integration sites and vector integrity for clonality and genetic stability. And we provide services, but we also provide kits. This is a recent addition to our offering portfolio. So if you would like to analyze a few samples per year, you can just send us the samples, and we provide an all-inclusive service starting from 2,650 euro per sample, with a standard turnaround of 8 weeks. But if you would need your results a little bit faster, we also offer expedited services that can release the data within 4 weeks. But of course, that has an additional fee. For companies interested in a very high throughput processing of samples, we also offer kits that allow complete TLA to perform the complete TLA protocol in-house. It can be automated or manual, depends on the needs, and we are working also on a software analysis package, a click-based, that would allow any person who is not a bioinformatician to also analyze TLA data. That will be released hopefully later this year. And when you run TLA in-house, well, you can have your results faster, and also it means a significant reduction in the cost per sample. So this is the implementation is something that some of our larger customers are also very interested in looking at, and we can provide support to establish the protocol in-house. Please feel free to contact us if you would like more information about this. And one interesting new offering is that we are now have the service that takes care of everything related to sample pickup and shipment. So we can make your life easy also when sending the samples to us from any place in the world. This is an optional service and takes care of everything, including the box, the dry ice, the import documentation. And we have a flat price for Europe and another flat price for any other other place outside Europe. And yeah, not everyone has a courier deal. So for some of our customers, they are very happy that now we can include this, and it means less administration work for them. So we can help arranging the shipment of the samples. Well, I hope you have obtained today a good overview of what we do at Surgentis and how we can support our customers working on cell line development projects. And before I conclude, I would like to tell you more about our company. Surgentis was founded in 2012 as a spin-off from the Hubrecht Institute and the Dutch National Academy of Sciences. And we are based in Utrecht, the Netherlands. And as you already know from the talk today, we provide services and kits focusing on our TLA technology. And this is part of our team. This is the commercial team and the service team. Of course, there are way more people working in the lab and in the data analysis, but these are the people with whom you will normally interact when starting a project. And for cell line development projects, I am the sales and account manager helping in setting up the initial details of the project. And my colleague Cheryl, she's the scientific account manager in charge of the interpretation and delivering the final reports. And let's talk. If you will need support and you are interested in performing TLA as part of your project, feel free to contact us through our different channels. It can be via email or via LinkedIn, via the website. We would be very, very happy to support your work, so please feel free to contact us. And with this, I would like to thank you for your attention, and please let us know if you have any questions, send us a message. We will be very, very happy to help and provide further information that might be necessary. Thank you so much, and bye-bye.